Hi, Louis. You can start. Thank you.
Thank you very much. Okay, so hello everyone. Welcome back to day 2 of the Ensemble Plans browser workshop. My name is Louise and I will be delivering today's workshop. So let's just go through the workshop agenda. Um, so yesterday we covered an introduction to the browser. So I've shown you how you can navigate the browser, what the homepage looks like. And I also showed you the different species that are available in Ensemble Plants. Next, we looked at how we can visualize genomic regions in the browser, so how you can view or search for coordinates in Ensemble Plants. And today, I want to continue on and show you the different genes and transcripts pages on the browser. So if you remember from yesterday, I did a presentation explaining how the data is um, being processed in Ensemble. And then we moved on to a demonstration. And the first bit that we did was exploring genomic regions. So today I want to show you how you can actually look for a gene or a transcript on the browser. So let me switch to the demonstration slide just as a reminder. So we looked at this gene in Triticum aestivum, which is wheat, and we visualized the region. So next, we want to find information about the gene and its transcript. So if you would like to follow along, I will share the gene name with you in the chat. Let me just find my chat window. Here we go. So this is the gene that we are searching for. And what we're going to do is we're going to switch to the browser and look for the gene. So this page is the training page. Don't worry about this for now. I will share the link again once we start our exercises. So I'm going to switch to my Ensemble Plants browser. And you want to go to plants.ensemble.org. I will paste that link into the chat as well. So plants.ensemble.org, ensemble without the E at the end. And once you load the page, you land on the home page. So this is the home page. We explored this yesterday. So today we want to search for our gene of interest, which is in wheat or Triticum aestivum. And you can enter your gene. Oh, that's not the gene. Just copy the gene again. And you can enter the gene just underneath the drop-down menu. So in this green box here, and then you can click go. And when you get something like this, that just means that it didn't connect quite right. So let's just hit the refresh button. Okay, let me just try that again. Okay, so it seems like there is currently a site-wide downtime. Sorry about this. I'm just gonna check with my colleagues if they're doing some kind of work with Ensemble at the moment. Okay, so seems like there's intermittent issues. So all you need to do in this case is to refresh the button. So there's intermittent server issues, connection issues with our servers right now, but it should work okay now. Okay, so let's start again. So first we need to select our species of interest, which is Triticum. And you can enter your gene of interest underneath in the text box. And then you can click go. Once you click go, you get results searching the Ensemble Plants website for your term of interest. So that's the gene here. And if you want to look at gene information, you want to click on the gene ID. But if you want to look at the genomic region, so if you want to visualize the genomic region, you want to click on the coordinates. We did that yesterday. So now we're going to click on the gene ID. And if you look at the top of the page, so we know which species we're in. This is Triticum. And if you look underneath, we have these different tabs. So yesterday we explored the location tab. This is when you want to look at features in the genomic region of the coordinates you search for. So in our case, this gene in wheat. And now you also notice we have this gene tab. So if you want to find any information about the gene, for example, the function of the gene, you need to switch to the gene tab for that. By default, on the left-hand side, you can see that we land on the summary page. So that's what you're seeing here on the right. And in the summary page, you get an overview of the gene. So at the very top, we can see this is the gene ID. We've got the location. The gene is located on a reverse strand. It has one transcript. So remember, a gene can be transcribed into multiple transcripts or splice variants or isoforms, as we call them. We call them in Ensemble, we call them transcript. So in this case, for this gene, there's only one transcript. We've got 186 orthologs, which we will explore a bit more, talk more about later today, and 5 paralogs. If you open the transcript table, so if you click on Show Transcript Table, you can see, oh, there we go. Sorry about that. If you open the transcript table, you can see the transcript, the different transcripts of the gene. In this case, it's a very small table. There's only one line because we only have one transcript. But let's focus on the gene information for now. So I'm going to close it back up. Under summary, you can find what type of gene this is. So this gene is protein coding and the annotation method. So this gene in particular was annotated by the IWGSC, who provided the genomic sequence and did the annotation of the genes. Scrolling down, you have a region in detail view. So this is what we looked at yesterday. And our gene of interest is highlighted in green here that I'm pointing out with my mouse. So this entire feature is our gene ending in 500. And just like in the location tab, this is interactive. So you can click on the feature, find more information about the feature, find the coordinates, what type of gene it is, and so on. Okay, so let's explore the different pages that we can find for this gene. So one of the pages I want to show you is the sequence. So if you click on sequence in the left-hand panel, and if you scroll down, we have our sequence in FASTA format. So we know it's in FASTA format because it starts with a greater than sign, followed by the FASTA header, followed by the sequence itself. So you'll notice at the top, we've got a legend, and there are different regions highlighted in different colors in the sequence. So any sequence that is colored in red font, these are the exons of our gene of interest, so the gene we just searched for ending in 500. And anything that's highlighted in peach is any other regions, any other exons, sorry, in the region. So in this case, we're only seeing, well, up until down here, we're only seeing exons of the gene ending in 500. But if you scroll to the bottom, you can see you have this bit of the gene that is not red, but it is highlighted in peach. And that's just a neighboring gene. So by default, we're showing you the flanking regions of the gene as well. So these are the neighboring sequences of the gene. So that's why you're seeing a bit of the neighboring gene down here. And you can change this. So if you want to see the sequence of your gene only, you can go to configure this page on the left-hand side. And you can change the number of flanking sequences up and downstream. So we show you by default 600 bases up and downstream, but you can change this to 0 so that you're only visualizing the gene ending in 500. And there are other options that you can change in this menu as well. So if you want to change the number of base pairs per row, you can do that from this drop-down menu. You can also visualize any variants in the sequence. You can add some line numbering. So this is always useful. It helps you find in which, what number, what nucleotide you're actually looking at. And there are also different filters that you can highlight if you're if you are visualizing any of the variants. So let me close this just to show you how it would look. So I've deleted the flanking regions, flanking sequences, and I've also added the line numberings. And you can see if you look at the entire sequence, you don't have these flanking regions anymore. So there's no neighboring genes here anymore. And you also have these line numbering. So the sequence starts at number 1 and ends in 2761, 2, 3, 4, 64. This view is also very useful if you want to perform a blast search. So you can highlight a specific region in the sequence with your mouse. And when you let go of your mouse, you get this pop-up window that says blast selected sequence. And that will search any sequence, any similar sequence in other species. You can also download the sequence. So if you click on the download button just above the FASTA file, you get another pop-up menu, and you can select what format you want to get. So you can select from FASTA or rich text format or RTF. And you can also select what type of sequence you want to get. So do you want to get the cDNA? Do you want to get the protein sequences? Do you want to get the intronic regions only maybe? So there are different options you can choose. And if you scroll to the bottom of the menu, you get a preview of the different file formats. So if you've forgotten how these file formats look like, we give you a little preview. So we've got the FASTA format, which looks like this, and the rich text format, which looks like this. So the main difference is that the FASTA format is just a text file, whereas the rich text format includes the different colorings that you're also seeing in the web browser. So you might want to use rich text format if you want to visualize the sequence in a report, for example. And the FASTA format is usually the standard format for any downstream analyses if you're doing any later on. If you want to exit the menu, you can just click on the check icon in the top right-hand corner, or you can click anywhere outside the menu, and that will get rid of it. Okay, so this is the sequence. Let's look at some other options that we have. You can also visualize any variants in the gene. So if there are any genetic variants that are found in the gene, you can visualize it by clicking on variant image. And this is our gene of interest, so this very middle line here. And you can visualize any variants, if you scroll down, any variants that are found in the genome. So there are quite a few in this gene here that you can see. And there's different types of variants as well. These are colored in different colors. And we will explore this more in the variation module tomorrow as well. And then you've also got, what else did I want to show you? Oh, yes. You've also got a link to external references. So if you wanted to find out more information about the gene in other databases, for example, who might have more information, even more information, you can go to external references here in the left-hand panel, and we can see that there is an entry for the gene in Netminer wheat and also in wheat expression. So this is showing you any expression data from that database. Okay, so this is gene-based information. If you want transcript-based information, so for example, if you want protein information, you need to switch to the transcript tab. And because there's only one transcript for this gene, it's already open here. But if there are any other transcripts, we have these listed in the transcript table. And for each transcript, we have different information. For each transcript, we have different information available. So we've got the transcript ID, we've got the length in nucleotides, in bases, we've got the length. If it's a protein coding gene, we've got the length of the protein, what type of gene it is. If you see anything underlined like this, you can always hover over with your mouse, and that gives you a description of this feature. And we also have a link to UniProt, which is another database that you can where you can look up information about proteins. We also have this column called flags. This is more useful, especially for the Ensemble main site, which is dedicated to vertebrate genomes. And this is just the different tags are just highlighting how confident we are in the annotation. Now, because the annotation for wheat was imported from the IWGSC, the only flag we've got here is the Ensemble canonical flag. And you can, again, hover over it with your mouse if you want to know what that means. Okay, so let's look at transcript information. So I'm going to click on this transcript ID, or alternatively, you can also click on the transcript tab. And if you focus on the left-hand side again in the panel, you can see that these have changed. So there are now different displays available to us. So before we had the sequence display, but now we also have exons, cDNA, and protein sequences. So if you wanted to visualize these, let's look at how this differs from the gene page. So I'm going to click on the cDNA link. And if you go to the right-hand side, we have again our sequence, but this time this is not in FASTA format. We don't have the FASTA header here. This is just the sequence. But you'll notice that this is actually made up of three different rows. So the first row is showing you the cDNA. The second row is showing you the coding sequence, so CDS. And then the third row is showing you the protein the amino acid sequence. So a gene would usually start with an ATG, which codes for methionine. So that's what you're seeing here. M for methionine, and then all the other amino acid codes. Here as well, you can configure the page. You can change any options that you might want to. You can highlight or visualize variants in the sequence. You can change the line numberings and so on. You can also, so if it's a protein coding gene, you can also visualize the predicted protein model. So we have the PDB 3D protein model, and we also have the AlphaFold predicted model. In this case, for this particular protein or transcript, we only have the AlphaFold predicted model. So if you click on the AlphaFold link on the right-hand side, we've got our amino acid sequence at the top, and underneath, we've got our protein structure. And the protein structure is interactive, so you can use your mouse to move around, you can zoom in and zoom out. And if you look to the right-hand side, you can also see how confident the model is. So there are certain areas in the gene, in the protein, where the confidence is a bit higher or lower. You can also highlight any specific regions in the protein. So any exon, you can highlight any exons in here. You can also highlight any specific features from PFAM or Panther, so looking at different protein domains and so on. And if you want to download the image, there is an option just in the right-hand corner of the protein model. There's kind of camera capture image icon, I guess. And if you click that, you can select what format you want and download it straight to your computer. Another thing I want to show you as well are any external references for proteins. So here you can find under general identifiers, you can find links to external databases that are looking at this protein, where you can find even more information about the protein, like domains and so on. Okay, so this is pretty much all I've wanted to show you for this part. So we have time for exercises now. So if you go to the training page, which I will also share in the Zoom chat. If you go to the training page and scroll down under demos and exercises, please feel free to start with either of these. So you can start with either exploring genomic regions or genes and transcripts. Again, exercises are just underneath. We've got a few different options that you can explore. Please know that you're not going to have enough time to complete all of them. So just try to maybe do only one exercise from exploring genomic regions and another one from genes and transcripts. This training page will be available to you indefinitely. So that means that if you have a bit of spare time later today or tomorrow, you can always do the exercises yourself then. So I'm going to give you 20 minutes for these exercises. And if there are any questions in the meantime, please ask away. And then we're going to come back together at, sorry, what time is it now there? So we're going to come back together in 20 minutes time and move on to the next module, which is comparative genomics. So it's 7:24 now. Okay, let's say 7:45. Okay, welcome back everyone. So it's 7:45 Indian Standard Time now. Hopefully you were able to do at least some of the questions and the exercises. But if you didn't have enough time, please know that the exercises will still be there later on. So you can always come back to them and try to do them in your own time. I think I've answered all of the questions that came in. Thank you very much for all of those questions. Keep coming in. I'm very glad that you're asking questions. This is really good. There are a few questions that I need to do some research on, so I've asked the participants to add them to the living document. But if you have any questions, let's say maybe after today's session, you can also enter them into the living document. So let me share the link with you. I've shared a link with you in the Zoom chat. You can enter your questions into page 5 if you have any outstanding ones after the workshop today, and I'll try and get through them before tomorrow. Are there any other questions? All right. In that case, let's move on to our next module. And hopefully I'll be able to get through the demonstration for this as well. So let's now look at the comparative genomics data that we have available in Ensemble plants. So we're going to look at two main themes in this module. I'll talk about the applications of comparative genomics and the species that are available in Ensemble. And I'll introduce you to the comparative genomics data that we provide at the gene level. So these include gene trees and homology predictions. And then we're also going to look at whole genome alignments, or WGAs, for which we have pairwise and multiple alignments. And I'm also going to mention shared symphony and what this is and how you can find it in Ensemble. So let's look at the applications of comparative genomics. With sequencing data being generated with a much faster and cheaper rate, a lot of previously unavailable genomes have now become available, meaning that we can start making comparisons between species which we didn't really have access to before. So researchers in the field of comparative genomics are especially interested in examining the evolutionary relationships between species, finding and understanding the differences between the genomes, and so on. So a more practical application of comparative genomics is to find a gene's function based on homology. And this is especially useful for a less well-studied genome where only very little or no low quality annotation data is available. And then finally, you can also identify any highly conserved regions within genomes. And there are many different ongoing projects that generate sequencing data, for example, for researchers to do comparative genomic analyses on. One big sequencing project here in the UK is the Darwin Tree of Life, which is aiming to sequence all of the living organisms in Great Britain and Ireland. In Ensemble, we perform comparative genomic analyses by taxa. So this means that we compare all vertebrates against one another. We compare all plants against one another, all metazoa against one another, and so on. We don't do these on the bacterial species simply because there are too many bacterial genomes that we have in Ensemble. I think we have about 50,000 genomes available right now. You can find the comparative genomics data under the Gene tab, which I will show you during the demonstration. And whenever you see the word compara in Ensemble, this just means comparative genomics. So we've just shortened it. So if you hear me say compara, I mean comparative genomics. We also perform pan-taxonomic comparative analyses, so comparative analyses across different taxa. So for this, we take a number of representative species from each taxon. So we take some species from protists, some species from plants, fungi, metazoa, vertebrates, and some bacteria. And then we perform comparative analyses on all of these representative species. And again, you can find any pan-taxonomic comparative analyses in the gene tab, which I will show you after this presentation. We can divide the comparative genomic data and ensemble into two main types. So we've got gene-based and sequence-based resources. Under gene-based resources, which you can find under the gene tab, these are phylogenetic tree and protein families. And for the sequence-based resources, which you can find under the location tab, these are whole genome alignments, so WGA, whole genome alignments, which helps us infer any ancestral sequences, any statistical information such as conservation scores across different elements. And this is also where you can find any synteny information. A type of comparative analysis data that we produce are gene trees. So for this, we take a representative protein of each gene from each species. So that's why we also call them protein trees. And then we classify the representative proteins into families using different clustering methods. We align these, and then we reconcile them against species trees to produce the gene tree. And then finally, from the gene trees, we can make our homolog predictions and even potentially infer the function of a gene in a different species. So in Ensemble, there are two main types of homologous relationships that you can find information on. And these are orthologs and paralogs. So here on my slide on the right, we have a simplified phylogenetic tree, or phylogeny, what we call, to just explain these relationships between the genes a little better. So these nodes here, the end nodes here that you're seeing, these are genes in different species. So this is one species, this is one species, and this is another species, and this is a third species, and they all evolve. All these genes evolved from the same ancestor. So that's what you're seeing here. So if we look at the different types, orthologs are genes that have emerged through a speciation event. So these are the ones the nodes that are colored in blue on the phylogeny. So for example, C1 and B1, we have a blue node here. So there was a speciation event at this point in the common ancestor, and then the gene became C1 and then B1. So C1 and B1 are orthologs of one another. And they can be described as one-to-one orthologs or one-to-many orthologs. If we look at our phylogeny again, C1 and B1, the ones that we just looked at, are one-to-one orthologs because they are orthologs only to one another. Whereas one-to-many orthologs would be an example for this node here. So we've got A, the A gene in this species, and then we've got B1 and B2. So A has two orthologs in the species, which is B2 and B1, so one-to-many. And the second type of homologous relationships are paralogs. So these are genes that have emerged through a duplication event. And these are represented as this red node in the phylogeny. So for example, if we look at C1 and C1 and C2, and also B1 and B2, So these are paralogs of one another. B1 and B2 are paralogs, and then C1 and C2 are paralogs. And these have emerged through a duplication event. And often when orthologs, when paralogs, when homologs emerge from a duplication event, they would evolve new functions. So we've got two homologous relationships. We've got orthologs and we've got paralogs. Orthologs are genes that have emerged through a speciation event, and then paralogs are genes that have emerged through a duplication event. In plants in particular, we also have homeologs. So these are just pairs of genes in a polyploid genome that have undergone hybridization events. So wheat is a domesticated plant and an example of a polyploid genome, and it underwent hybridization events where basically three species have been merged into a single organism. So this increased the ploidy, or the number of chromosomes in the nucleus. And this is quite common for domesticated plants, as you probably know. It also happens to different fruits and vegetables. So here we have another phylogeny and an example of homeologs. So homeologs are genes that were orthologs to begin with. We have these three wheat species or wheat genes, we have three of these wheat species, each with their own ortholog. And when they hybridize, becoming ABD, all these three genes are now found in this new genome, in this polyploid genome. So this is an example of a homeolog or homeologs. For any homolog information, you can find these under the Gene tab as well in the browser. There are also different representations of gene trees. So far, we have looked at gene trees, but you can also find gene gain and loss trees in Ensembl. So these trees just show the same information, but with a focus on what genes were gained or what genes were lost across time. Gene gain and loss trees are a more simplified or collapsed representation of the evolutionary history of a gene. Okay, so for our demonstration, we are going to look at, we're going to continue on with our wheat gene ending in 500. And this time I'm going to show you how you can find information about gene trees and its homologs and also homeologs. So if you want to follow along, I can paste the gene name into the chat again. And you can copy that. And what I'm going to do now is switch to my browser and go back to the Ensembl website. So this time we want to go to the Gene tab. So first I'm going to show you how to find gene trees and homologs and homeologs. So if you switch to the Gene tab, in the panel on the left-hand side, we've got plant compara. So this is comparative genomics within or between plant species only. And we also have pan-taxonomic compara. And these are comparative analyses across different divisions. So plants, vertebrates, bacteria, and so on. If you see any link grayed out like this, so if you can't click on the link, that just means that the data is not available. So I believe Triticum aestivum, wheat, in this case, is not part of the pan-taxonomic compara, because for this type of analysis, we're only taking a number of representative genomes from each division. But we do have plant compara available. So if you want to look at the gene tree, the phylogeny of this gene ending in 500, you can click on gene tree. And on the right-hand side, we've got our gene tree and some summary statistics or some summary numbers. So each gene tree that we produce has its own ID. That's what you're seeing up here. We can also see the number of genes in the tree, the number of speciation nodes. So these are orthologs, number of duplication nodes, paralogs, and number of ambiguous nodes, and number of gene split events. If you scroll down, we have our phylogeny, which is already quite large. But you'll note the first thing you'll notice are these triangles. And what these triangles mean is that we just collapse that node to make the phylogeny a bit more easy to read for you. So if there is a specific genus that you're interested in, kingdom. Sorry, let me double check that name. Kingdom, I think of. Oh, genus. Yeah, no, I was thinking of the genus. If there's a specific genus that you're interested in, you can expand the node by clicking on the triangle. This gives you this pop-up window, and then all you need to do is select expand the subtree. And you'll notice the tree has gotten much bigger and it is a bit more difficult to read. So this is why we try to collapse some of the nodes. So our gene of interest in wheat is highlighted in this green font, so we can see that here. And on the right-hand side, this is a visualization of the alignment, so the sequence alignment of the protein. So anything that is empty, this means that there is a gap in the alignment, so there's no alignment there. And then whenever you see a green shade like this, or these are all aligned sequences, so aligned regions in the gene, in the protein. If you scroll down, you can find out the legend. So we've highlighted the speciation nodes, the duplication nodes. You can also see what the collapse nodes mean, what the colors of the collapse nodes mean. And we also have a legend for the alignment itself. So there are two main colors. So, yeah, two main colors in here. So we've got the dark green and the light green. The dark green just means that there is a higher or more alignment between the sequences compared to the light green regions. This view is also downloadable. So if you want to include the phylogeny into your reports, you can export the image and you can select what format you want to get. Or if you want to download the data of the phylogeny, so if you want to download, for example, the alignment, you can click on export and select your data format. So depending on what you're going to do with the data, you might need it in a very specific format. We have different ones available to you here. But if you're not sure what these look like, here we have a reminder underneath an overview of each of the different formats. And then you just need to select what format you want, and then you can download it to your computer. All right. So this is the gene tree. We can also look at orthologs, a list of orthologs in the gene, of the gene. So again, if you go to the left-hand panel and click on go to Plant Comparer, and then click on orthologs, you get a list of all of the orthologs available for this gene ending in 500. So if there's a specific species set you're interested in, you can select that from this table here on the top. But by default, we're showing you all of the species, so all of the available plant species in this browser. So let's say you are interested maybe in any orthologs in barley, for example. So you could use this search filter in the top right-hand corner and search for barley, for Hordeum vulgare. In this case, if there is no entry like it is here, that means that there is no ortholog available. So you can double check that in this list at the bottom. So indeed, there are 8 species that don't have an ortholog with this gene ending in 500, which includes Hordeum vulgare, which is barley. Okay, so let's look at an example of an alignment. So maybe we can look at the ortholog in Arabidopsis. So if you just go through your list, there are many orthologs in Arabidopsis. Actually, you can see there are multiple entries, 1, 2, 3, 4 orthologs in Arabidopsis. And there's many to many orthologs. So that means that there are multiple orthologs in wheat to Arabidopsis. But what I want to show you here is that you can actually compare the different regions, so you can visualize the alignment between the 2 species, and you can also visualize the sequence alignment between the species. So there are 2 options. You can look at the protein alignment, or you can look at the cDNA alignment. You just select the protein alignment. Okay, so we've selected the ortholog alignment between Arabidopsis and then our species of our gene of interest in wheat. So this is the alignment that you're seeing down here on the right. This is what we call the clustral W format. And we've got our reference, so our wheat sequence at the top, followed by the Arabidopsis sequence underneath, followed by some scoring symbols. So these different symbols, the asterisk, the colon, and the dots, they all mean different things. If you're not familiar with the clustral W format, or if you don't remember like me, you can always click on the question mark here under next to ortholog alignment. That will open the help page for that alignment, and it will tell you what the different symbols mean. So if you see an asterisk like this one, this means that the nucleotides between the 2, or the residues between the 2 alignments are identical. Then we've got colon, which means that the function is conserved, and then a dot means it's semi-conserved, and a space would just mean that there is no conservation of function. So to help you understand the alignment a bit better, you can, instead of looking at the individual protein or amino acid residues, you can just look and focus on the symbols at the very bottom. Okay. And then final thing, you can also look at homologs. So if you go to homologs on the left-hand side, in this case, there's only this one gene or this one homolog in wheat, but often you would find that you have the gene as well in other chromosomes. But for this example, there's only. Okay. Oh, there is a question in the chat, and the question is, for the gene tree under Plant Compara, apart from the gene of interest being highlighted in red, there are other genes highlighted in pink. What are these? Okay, so let's have a look. So I'm back in my browser. I'm going to switch back to the gene tree and try and find the other colors. Okay. So For example, we've got Arabidopsis here, and we've got other gene here. Oh, that's an interesting question. I'm not actually sure why that is different. My guess would be for this case is that Arabidopsis is a very popular species to study. So often people would want to look at the gene in Arabidopsis as well. So we're just um, highlighting that for people to find it a bit easier. But I will need to double check that because I'm not 100% sure myself. So if you could add that question to the living document, that would be really useful. And then I will get a confirmation from my colleagues to see if that is indeed the reason why we've colored that. Thank you. Okay. Any other questions? All right, in that case, let's continue with the presentation. That wasn't it. So in the first half of the module, we looked at gene-based comparative genomics data available, but we also have whole genome alignments available. And these are especially useful if you want to identify any highly conserved regions in the genome. So often if you have a highly conserved region in the genome, this would mean that the genes located in that region are still or likely functional. So they've evolved very slowly and have a function. But they can also include both coding and non-coding sequences. But whole genome alignments are also useful to support any problematic gene predictions. So any regions in the genome that are difficult to predict genes on. So for example, highly repetitive regions in the genome. And whole genome alignments are also very useful for synthetic regions, so to define any synthetic block in the genome. In Ensemble, we have two types of whole genome alignment. We've got pairwise and we've got multiple whole genome alignment. So pairwise alignments are performed in pairs, as the name says. So this is when we take two genomes and we align them against one another. In Ensemble Plan, we have a number of these pre-computed alignments available. So we have pairwise alignments of Arabidopsis against many other species, as mentioned, because it's a very popular species in research. But we also have pairwise alignments available for rice and rice cultivars, and also grape. And we also have alignments available between the different wheat components, so the different chromosomes. And if you want to find out if your gene of interest has these alignments available, your genome of interest has these alignments available, there is a link at the bottom of my slide, and that brings you to the documentation page with a list of all of the different pairwise whole genome alignments we have. We also perform multiple whole genome alignments against predefined species sets. So these are species, a group of species that we have defined beforehand, and we perform alignments against each of them, against all of them, against one another. So these alignments, instead of doing one genome against one other genome, what we do is we take all genomes from the set and we align them against one another. And there are different multiple alignment analyses that we have available in Ensemble. So in Ensemble Plants, we've got the EPO analysis, and this includes 8 rice species / cultivars. And the second multiple whole genome alignment we have is the EPO extended. So these include a larger number of species. So instead of 8, it's 11 species. And the main difference here is that we've included some more fragmented assemblies in this alignment pipeline. So, for example, any genomes that don't have as high quality as the other genomes here. Next, I want to talk about synteny. So synteny is the conserved order of aligned homologous block between species. So what you're seeing on my slide is an alignment between Arabidopsis and rice. So let's say we've aligned this region in Arabidopsis to the rice genome, and we have found a number of regions that align between the different genomes. So anything that's shaded in green, these are the regions that have aligned to the rice genome. But if you look closely, I'm not sure if I can actually zoom in. Let me check. Ah, brilliant. So if you look closely at the Arabidopsis region, this has 4 genes found in this block, in the synthetic block. And these are ALG11, UTP, NEC5, and NEC3. Now, if you align this block to the rice species, we can see that the order of the genes actually remain the same. So it's ALG11, UTP, NEC5, and NEC3. So this is what we call a synthetic block. It just means that the order of the genes of the homologous block is conserved across the different species. And this can also be in reverse, so it doesn't have to be in the same direction. So the second example here, we've got the THS gene, VP, and CK. And if we align that region in the rice genome, this is actually reversed. So first it's CK, and then VP, and then TH. I hope that makes sense. But if you have any questions at all, please let me know. Okay. So if you're interested in the more technical side of the comparative genomics analyses that we do, so what pipelines we use, what algorithms we chose, and why, there is a very interesting paper here on my slide that you can refer to. All right, so let's look at this region in wheat, and we're gonna go to the location tab and find alignments and find any conserved regions within the gene. So for this, I might need to jump out of my presentation and actually copy the coordinates. Right. And I'm going to paste that into the chat if you would like to follow along. So let's switch to the browser. And we're going to go to the very top of the page. And I'm going to go back to the home page and start a search from scratch. So we are interested in wheat. So we're going to select wheat from our drop-down menu, Triticum aestivum, and then we've got our coordinates. So these have to be in a very specific format. They start with the chromosome. So 6b is the chromosome, followed by a colon, followed by the start position or start coordinate, hyphen, and coordinate. And then you can click go. And it sends you straight to the location tab. So for any whole genome alignment information, you want to be in the location tab. For any gene trees or homolog, you want to be in the Gene tab. We are on the region in detail page, so that's what you're seeing here on the right-hand side. Just as a recap, we've got our chromosome overview, we've got our region overview, so our region of interest is highlighted or highlighted in this red box here. And then we've got our region in detail view. So this is only the region that we just searched for. And in this region, we can add tracks to visualize any alignments in any conserved regions. So under you can click on Add, Remove Tracks, or configure this page in the left-hand panel. And that gives you a pop-up menu. And on the left-hand side, we want to open the comparative genomics category. So here, the first few options we've got are pairwise alignments. So we've got a number of alignments. If you want to visualize, for example, an alignment between the different wheat cultivars that we have available, you can highlight that. So let's just select a few of them to look at how they differ. So I'm going to select the Arenal 4, and I'm also going to select the Julius cultivar. There's also, oh, that's actually it. Okay, so we only have pairwise alignments available in this case. Yes, because wheat is not part of the pan-taxonomic compiler. Okay, so let's close this window, see how it looks on our browser, see how the track looks. So this is the region in detail view. We've added 2 tracks. We've added the track for the Arena cultivar, and we've added a track for the Julius cultivar. So what you could do here is you can compare the 2 cultivars and see how they differ. So for example, if we focus on Julius, anything that is highlighted or that is shaded in pink, these are aligned regions. And then if it's empty like this is a gap. And then we have another alignment region here. So they are quite similar to each other, but we can see in the Arena cultivar, there is a small gap in this region of the genome. So what you could do is look at what this region actually overlaps with. So I can see this actually, this might even overlap with a variant. So maybe that could explain why that differs. Sometimes it also overlaps over genes. So you could look at whether it overlaps any intronic regions or exonic regions and so on. And don't forget that you can always zoom in and out of this in this view if you wanted to see more details. I'm not going to do that here right now, simply because I know it will take some time to load and I want to show you some other things as well. But please feel free to explore this in your own time. If we go back to the top of the page in the left-hand panel, we have a whole section for comparative genomics. So here you can align different regions of genomes in more detail. So you can do a text alignment. This would be what we saw earlier between the ortholog in wheat and Arabidopsis. And then you can also align it as an image. So this would put the 2 genomes on top of each other and align it as we saw in my final slide there in the presentation. Or you can also view the synteny. So let me click on synteny. We've got our chromosome overview up here. Our region of interest is highlighted in this red line here. It's a very small region in the chromosome. And this is the synteny view. So the middle chromosome is our chromosome of interest. So that would be the 6b chromosome. And what you need to do is first select your species of interest. So let's say we want to compare it with Triticum turgidum. So a different triticum is a different triticum species. And then you can change the chromosome if you like, but I'm just going to select a different species. So we've got our Triticum aestivum chromosome here in the middle. And on the side, as a smaller chromosome, we've got the Triticum turgidum chromosome. And any regions that have any synthetic regions, so any blocks of in the genome where the order of the genes are kept or are conserved, these are highlighted in green. So we can see most of the genome really is the order of the genes are conserved. And we have two different colors of lines. So we've got the red, which means that the conserved block is reversed. And anything that's a black line, this means that the order is the same, so in the same direction. All right. And down here, at the bottom, you can find more links to the region comparison. So you can compare the two regions on top of each other as an image to look at the different features that you can find in that region. Okay, so that's all I've wanted to show you for the comparative genomics part. We only have a couple more minutes left, so we're not going to have time for exercises, unfortunately. So what I suggest is that you can try the exercises on your own time. And tomorrow, when we come back together, you can ask questions if you have any. And if not, we'll just move straight on to the variation module. So this is where we talk about the different genetic variation data we have available in Ensembl. In the meantime, I can see there are a few questions here. So one question is what is last Z alignment? So when you do short sequence alignments, usually you use the tool called BLAST. And last Z is just a more recent version of that. So with a different kind of algorithm. So that's what last Z is. And then the next question is comparative genomic analysis will be the same for bacteria, whether we can we find any complete genomes for analysis and use the Synteny to identify similar genes in the bacteria. So unfortunately, we don't have comparative genomics analyses for bacteria against all bacteria, simply because there are too many bacteria available there. But there are ways to do it yourself. And if you want to find, or if you are interested in doing some courses that talk you through how you can do comparative analysis on bacterial genomes, you can enter your question into the living document, and I can send you a few links for that. There are some very useful ones that you can check out. The next question is, in case of Synteny, we are considering conserving the order of the genes. However, are we at the same time taking the directionality of the strand into account? Or is it considered regardless? Yes, so it's regardless. So synthetic blocks, no matter whether they're in the same direction or in the opposite direction, we still call them synthetic blocks. But in Ensembl, you can see whether it's in the same direction or the inverse direction by looking at these different colored lines. I hope that makes sense. Okay, any other final questions that you may have before we finish up for today? I know there's a lot of information to take in, but luckily all the sessions are being recorded. So if you'd like to go over anything again, please feel free to do so. And if you have any questions, please feel free to add them to the living document. I can see that there are already a few in there, so I'm going to go ahead and answer them after our session finishes here. But you can also ask them tomorrow at the beginning of the session.
Thank you, Louis. So I think all the questions are addressed, and it was a wonderful session, very informative. So
Thank you very much.
thanks for sharing your knowledge, and we will meet again tomorrow, same time.
Same
Okay,
time, same
so
link.
yeah, yeah, everything. Okay, so
All right.
I'm seeking your permission to close the session. Can I?
That's please go ahead. Yes, that's fine.
Yeah, thank you. Have some rest. Have a wonderful evening. Thank you.
Thank you, everyone. Have a good evening.
Yeah.